So probably as you all know already here in this wonderful seminar, we know the importance of the of the gut microbiome, in particular in health, and we know now that the important factor driving the variation and diversity in the gut microbiome is between industrialized societies and traditional societies, and this is due to the very different factors. Could be diet, could be antibiotics, pollution, exposure to other pathogenic organisms. And it's already known, and this has been published, that Latin America, as well as other regions, is highly undersampled and underrepresented in the microbiome world. And you can see from this map that essentially Latin America, and this was a set of 16S data set, is the least sample from all the regions. If we look at a different map where you can see scaled, this is the normal map and this is scaled to the sampling effort, you can see how this is heavily biased to the global north. So there is really a big gap in understanding the composition of the human gut microbiome in Latin American in the Latin American region. So this very large meta-analysis contained 168,000 samples, and only 1,000 of those were from Latin America, and all those were coming from just 11 studies. So they're very different reasons. So for sure, there are not only 11 studies coming from Latin America, there's in the order of hundreds of studies now, but there are different reasons why those data either are not deposited, or the metadata is not appropriate, or they're not in the right scale or the sequence type in order to be included in this type of meta-analysis. So in 2019, this Latin Biota Consortium study started, and it was led by Gregorio Iraola in Uruguay in the Pasteur Institute in Montevideo, in collaboration with the Sanger Institute with Trevor Lawless Group. And they have the aim to understand the composition and variability of the human gut microbiome within Latin American populations. It involved nearly 50 Latin American scientists from 8 different countries throughout Latin America that you can see in the map. Early collection efforts led to collecting approximately 600 samples. They included from the different countries, including adults or childs, mostly healthy, but some also presenting different types of diseases, and most importantly, with a very diverse rurality index. While some samples, like from Mexico City and Buenos Aires, were heavily industrialized, there are other samples from, for example, Maninaltepec and some samples in Ecuador that really were mostly rural populations. So the next analysis that I'm going to be showing you were mostly generated by Claudio Duran at the Sanger Institute. So here we're going to focus just on the healthy adults. We're not going to use the child with the disease for this example. So from Latin Biota, that means that we have 362 samples, and we collected other data sets that were publicly available for a total of 646 samples. We have some data sets where we only have like Chile, Bolivia, Ecuador, and Uruguay, where only Latin Biota is providing samples, but we were able to complement, for example, Salvador and Peru, where only public data sets were available. And we have a very nice split between urban and rural samples. So the first thing, this is all shotgun sequencing, relatively deep, 20 to 40 million reads per sample. So that allowed us to reconstruct a lot of different genomes. And what you can see here is a phylogenetic tree of all the diversity that we were able to assemble. And you can see very large diversity, for example, in this case, in Bacillora, and Bacteroides that are some of the most prevalent microbes in the gut. What you can see in these black lines are all the novel taxa that were not previously in GTDV-TK, and also some isolates that we have. So this is representing a great novelty. Then when we start doing the traditional analysis, alpha and beta diversity, what we can see here is that what is driving the most of the diversity is that split between rural and urban lifestyles. Even within Latin America, you can see that. And that lifestyle effect was even greater than the country itself. So there was common trends separating those rural samples from urban samples, even in between different countries. So we wanted to see what bacterial species were driving this difference. And we can say that there's several larger diversity in the rural samples and a lot of significant species, some that are present absent. So all these are present just in rural samples. And a lot of other ones that, even though they may be present in both rural and urban, they're highly enriched in the rural samples. As you can see by the colors, most of them are coming from the Bacteroidota. So we took the top 40 important discriminant features. We made a random forest model, and we can see that using only, I think, the top 100 species, we can make a 90% accuracy prediction by lifestyle, separating between rural and urban samples. So there was very interesting, this signature that was observed in Latin American samples that allowed us to differentiate between urban and rural. So then we wanted to see if using these samples could be applied worldwide. So since we have around 600 samples, we aimed at collecting more or less similar numbers on the different regions of the world. So these include a total of 3,318 samples from 5 continents and 38 countries. And this was a plot that we saw previously of the split between rural and urban, and using only those 100 discriminant features. When we map the data to those discriminatory features and we make the same plot, we see a very similar pattern where we see all the rural samples up here and then 2 big clusters of urban samples. Even if we color those by continent, we can see that this was the Latin American population, African population being mostly rural, Oceania population being mostly rural, Asia being both mostly urban, then Europe and North America being in this big cluster of mostly urban. So it's interesting that what we were able to use that data set to predict with 90% accuracy within Latin America, we were able to predict almost with the same accuracy the split between rural and urban samples worldwide. So this is very interesting, and because of time, I'm not going to go a lot deeper in the other analysis that we're performing, but this shows that there is a very significant signature of those bacteria that are being banished from urban settings. And they're not only present in rural Africa, but we also can find many of those species in rural Latin America. So where we are now, as I just showed, there are more than 600 samples from Latin America. In 2025, we will sequence another 200 from 3 different countries, and by 2026, we will be going to add an additional 700, mostly from Colombia. We are seeing a strong signal given by lifestyle change, stronger even than the signal by country or even by continent. And many of these bacteria that we're losing are common to different regions of the world. So now we're trying to understand a little bit of the metabolic capacities of those bacteria and what will be the important of losing those. And here is where collecting and preserving these bacteria is going to be quite important, because as we have seen this, we're losing those even from the urban settings within Latin America. So it will be very important to begin collecting and preserving those samples before they're completely lost, and while we get to understand what are the impact of those bacteria in the different settings. So to finish, I want to acknowledge all the Latin Biota Consortium, which I joined a couple of years ago, in particular the Trevor Lawley group and Claudio Duran, which he performed most of the analysis that I showed here. Pablo Tsukayama, who also is co-leading now this Latin Biota effort. And just for everybody, we're looking for Latin American partners interested in a second phase. So anybody interested can contact me through the email. So thank you very much.